Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the biomolecules, and the understanding of the biomolecule is very important for us to understand, for us to realize the importance of these biomolecules in running the several types of cellular activities. So far what we have discussed about the nucleic acids. So in that we have discussed about the DNA and RNA, and these two molecules are important for the information to be stored in from the one generation to another generation, and that's how they are actually going to relay the information from the one generation to the next generations. And subsequent to that, we were also discussing about the carbohydrates, and carbohydrates are mainly being required for energy production, and as well as in some cases they are also being part of the building blocks, where they are actually modifying the some of the crucial cellular factors and other things. So if you recall, in our previous lecture we discussed about the different types of carbohydrates. We discussed about the monosaccharides, disaccharides and polysaccharides. In addition, we have also discussed about the different types of structural as well as the biochemical details of these different types of carbohydrates. And the carbohydrates are present in the linear chain or to the cyclized form, and they are also showing the different types of isomeric properties. So with this brief discussion in the previous lecture, in today's lecture we are going to discuss about the how the carbohydrates are participating into the energy productions. So what we have discussed so far, we have discussed about the different steps of the metabolic reactions, what is happening into the glycolysis, what is the energy production within the glycolysis, and under the different environmental conditions, how the energy production is going to be modulated. And in addition to that, we have also discussed about the regulation of the glycolysis by the different means. Either it will be by the entry of the glucose into the cell by the mean of the insulin hormone, or by the covalent as well as the allosteric regulations of the different enzyme what is present into the glycolysis. Once the after the glycolysis, so from the glucose, once glucose is going to be utilized and it is going to be produced the pyruvate, this pyruvate is actually going to enter into the Krebs cycle, and the Krebs cycle is only be present into the eukaryotic cell. It is not present in the prokaryotic cell. So let's understand the next cycle, which is called as the Krebs cycle. So Krebs cycle, the Krebs cycle is named after the scientist who discovered the Krebs cycle, right? And the Krebs cycle is discovered by the Professor Hans Krebs, and it has the all sugar intermediate with the 3 carbons. That's why this is also called as the tricarboxylic acid cycle or the TCA cycle. So the Krebs cycle is also called as the TCA cycle, and it is being discovered by the scientist Hans Krebs. It is also known as the tricarboxylic acid cycle or the citric acid cycle. In higher eukaryotes, the Krebs cycle operates inside the mitochondrial stroma with the different types of enzyme. In the presence of the oxygen, the pyruvate formed during the glycolysis enters into the Krebs cycle for the further oxidation to produce the energy. So what you see here is the different types of reactions what is happening into the Krebs cycle. So this pyruvate, what you are going to see is it is actually going to be produced from the after the glycolysis. So once the pyruvate is going to be produced, it is going to be sent to the Krebs cycle, into the Krebs cycle. So it will enter into the Krebs cycle by the pyruvate will enter. First the pyruvate will enter into the mitochondria, and the when it enter into the mitochondria, it is actually going to be get converted into the acetyl-CoA, and the enzyme is pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex is a multivariate protein enzyme complex where we have the different types of enzyme, and it is actually going to utilize the coenzyme A to produce the acetyl-CoA, and this is a dehydration reactions, and what you see here is the first molecule of NADH is going to be produced in these reactions. Well, as soon as the acetyl CoA is actually going to be produced, it is actually going to combine with the oxaloacetate to produce the citrate, and the enzyme name is citrate synthase. So this is the first reaction and this is the second reactions. Now once the citrate synthase is actually going to produce the citrate is actually going to be utilized by the aconitase to produce the cis aconitase, and the cis aconitase is going to be further modified into the isocitrate. So and the enzyme is aconitase. So by cis, so in the second or third reactions, the citrate is going to be get converted into the isocitrate. Now isocitrate is going to be utilized by the enzyme which is called as the isocitrate dehydrogenase, and in this process again the another molecule of NADH is going to be produced. So this is the second NADH molecule which is going to be produced. The first NADH molecule is going to be produced when the pyruvate is getting converted into the acetyl CoA, and the second molecule of NADH is going to be produced when the isocitrate is getting converted into the oxalosuccinate. Now oxalosuccinate is actually going to go for the decarboxylation reactions, and that's how it is actually going to produce the alpha ketoglutarate by the enzyme which is called as the isocitrate dehydrogenase. Now once so there will be a production of carbon dioxide in this process, right? And once the alpha ketoglutarate is being produced, it is going to be get converted into the succinyl CoA, and the succinyl in this process again the NADH is going to be generated. So this is the third NADH molecule what is going to be generated, and the enzyme name is alpha ketoglutarate dehydrogenase, and again the another molecule of carbon dioxide is going to be produced. So this is the first molecule of carbon dioxide, this is the second molecule of the carbon dioxide. Now from the succinyl CoA it is actually going to be utilized by the succinate thiokinase, and the succinyl CoA is getting converted into the succinate, and in this step the one molecule of GTP is going to be produced. So instead of ATP it is actually going to produce the GTP. GTP is actually having the similar or the identical amount of energy what is present into the ATP. Succinate is getting converted into the fumarate, the enzyme is succinate dehydrogenase, and the one molecule of FADH2 is going to be produced. FADH2 is also going to be oxidized into the electron transport chain, but the only difference from the NADH is that FADH2 oxidation is only going to produce the 2 molecule of ATP from the 2 molecule of ADP, which means it is only going to produce the 2 molecule of ATP instead of the 3 molecule of ATP in the presence of in the case of NADPH. And then the fumarate is getting converted into the malate, the enzyme name is fumarase, and in this process again the one more molecule of NADH is going to be produced, and this is the fourth NADH molecule what is being produced within the glycolysis. And malate is again getting converted into the oxaloacetate, and that's how it with the help of an enzyme which is called as the malate dehydrogenase. Again the oxaloacetate is going to take the acetyl CoA which is going to be produced from the pyruvate, and that's how this cyclic reaction is going to be run for the another round. So what you see here is that we have the production of the NADH, we have the extensive production of NADH, we have the production of FADH2, we have the production of the GTP, and then there is a production of the carbon dioxide. So this carbon dioxide is actually going to be removed from the Krebs cycle, right? And how many molecules of NADH? You see that one molecule of NADH, second molecule, third molecule, and fourth molecule. So we have the 4 molecule of NADH, we have the 1 molecule of FADH2, we have 1 molecule of GTP, right? And if you see the pyruvate, so we are going to have the 2 molecule of pyruvate. So that's how, if you see, these molecules are actually going to be in the double the amount what is going to be produced. So let's see what is the ATP balance sheet of the Krebs cycle, because ultimately that is what we are discussing here, right? How the ATP energy is being produced from the carbohydrates. So ATP balance sheet, what you see from the Krebs cycle is very simple. There is no consumption of any type of energy. Like you remember that in the glycolysis we are utilizing the 2 molecules of ATP, and then only there is a production of 8 molecules of a net production of 8 molecules of ATP. Here there is no such investment, right? So what you see is the steps of Krebs cycle. So when there is a first step, when there will be a production of acetyl CoA, it is actually going to give you the 1 molecule of NADH, right? And remember that if the NADH is going to be utilized into the electron transport chain, it is actually going to give you the 3 ATP molecule, and that's why we have put the 3 into 1 molecule, right? So 3 ATP molecule is going to be produced in the production of acetyl CoA. Then in the step 3 there will be a generation of alpha ketoglutarate, and in that step also there will be a production of NADH, and that also is going to give you the 3 molecule of ATP. And then in the step 4 there will be a generation of succinyl CoA, and in this step also there will be a production of NADH, and that also is going to give you the 3 molecule of ATP. Then the step 5 there will be a generation of GTP, and as I said, GTP is actually going to have the same amount of energy what it is being found for the ATP, and that's how the 1 molecule of ATP is being formed. Then in the step 6 there will be a generation of fumarate, and there will be a generation of FADH2. So that is also going to give you the 2 molecule of ATP, because FADH2 when it goes for the electron transport chain, it is going to give you the 2 molecule of ATP. Then in the step 8 there will be a generation of oxaloacetate, and that also is actually going to give you the another molecule of NADH, and that's how it is actually going to produce the 3 molecule of ATP. Now what is the net balance for oxidation of the 1 molecule of pyruvate? So there will be a net production of 3, 3, 3, 1, 2, 3, and that is the 15 ATP molecule. But since the glycolysis is producing the 2 molecule of pyruvate, it is actually going to be the total production. So total production is going to be the 30 molecule of ATP molecule which will be generated. So which means if you start with the 1 glucose molecule, and if you consider that there is a enormous amount of oxygen present, then it is actually going to give you the 8 ATP molecule into the glycolysis, and it is actually going to give you the 30 ATP molecule into the Krebs cycle. This means it is actually going to give you the 38 ATP molecule, the net 38 ATP molecule under the oxygen, in the presence of oxygen. But if there is no oxygen present, then it is actually going to give you the, so what will be the energy production? If there is no oxygen present, then it is actually going to give you the 2 ATP molecule into the glycolysis, right? And it is going to see here, right? If there is no oxygen present, then this is not going to work. This is not going to work. This is not going to work, because there will be no electron transport chain which is going to be operational. This is not going to work also, and this is not going to work. So in this case, it is actually going to produce only the 1 ATP molecule, which means it is actually going to give you the 2 ATP molecule from the glycolysis as well. So that's why if there is no O2 present, then it is actually only going to give you the 4 ATP molecule. In fact, if the oxygen is not present, then actually that is going to make the Krebs cycle non-operational, because there will be no oxygen, so there will be no running of Krebs cycle, and because of that it's only going to utilize the glycolysis. So there will be only 2 ATP what is going to be produced if there is no oxygen present. That's why the amount of oxygen is actually going to decide how these how the cells are actually going to modulate their metabolism. So if there will be no oxygen present, then the pyruvate is what is being produced from the glucose molecule will not enter into the Krebs cycle, right? It will go and enter into the anaerobic oxidations, where the cells are actually going to produce the lactic acid, and they are actually going to produce the other derivatives of lactic acids. But apart from that, the Krebs cycle is extensively been involved into the energy production if the oxygen is present, and apart from that, the Krebs cycle is also being present in channelizing the different types of intermediates. So let's see the significance or let's see the how we can be able to regulate the activities of the Krebs cycle so that it is not going to it's so that all the reactions are under the proper control. So there are four rate limiting steps in the Krebs cycle and the point where it can be regulated. Okay. The step 1 is this, when there will be a conversion of pyruvate into the acetyl CoA, and what you see here is that these are the molecules which are going to be negatively regulated, which means if there is a very high amount of ATP, NADH, acetyl CoA or fatty acid, then that is going to inhibit this particular step. Whereas if there will be a very high quantity of ATP, NAD plus, coenzyme A, fatty acid or the calcium, it is actually going to amplify this particular reaction, or it is actually going to activate this particular reactions. So the first reaction is the conversion of pyruvate into the acetyl CoA is the first step which allows the entry of sugar moiety into the Krebs cycle. Pyruvate dehydrogenase complex is allosterically inhibited by the high ratio of the ADP to JDP or NADH to NAD plus and the acetyl CoA to acetyl CoA. See, this is because of this only, right? Because to require a very high ratio of this, it is actually going to give you the inhibition of this, which means there is a very high quantity of energy what is already been present in the cell. So if the energy is already present in the cell, why there is a need to run the Krebs cycle? So because of these molecules, like ATP is a energy molecule, NADH is also a energy molecule, it is actually going to inhibit the Krebs cycle. Then the second step is the first reaction of the Krebs cycle, which is catalyzed by the citrate synthase, and it is inhibited by the high level of NADH, ATP and succinyl CoA. So this is also the first step. This is the step also again which is going to be synthesized by the citrate synthase, and that is also been very regulated or the inhibited by the high level of these NADH, ATP and succinyl CoA. Then the third step is the isocitrate dehydrogenase, which is inhibited by the high level of ATP, NADH, whereas calcium and ADP stimulate this process. So this is the third step where it is actually going to be again modified or can be modulated. Then the fourth step is the alpha ketoglutarate, which is inhibited by the succinyl CoA and the high level of NADL plus, whereas the calcium stimulates this process. Now the Krebs cycle is not only for producing the energy. Okay, Krebs cycle is one of the central metabolic pathway which are actually being responsible for the Krebs cycle is connected to the acetyl CoA, and acetyl CoA is also connected to the fatty acids and all other kinds of pathway. So the intermediate what you see in the Krebs cycle are very much present in the many pathways what are present in the cell, and that's why the Krebs cycle is not only for the production of energy, it is also being utilized for the many other functions. So let's see what are these functions and what is the significance of the Krebs cycle within the metabolism. So Krebs cycle is the master regulator of the metabolism. So Krebs cycle is a centrally connected to the metabolic intermediates of the carbohydrate, proteins and lipid metabolism. It has several branching points where it can communicate with the other protein or the lipid metabolism. So what you see here is that the Krebs cycle, and it is actually been shown that what are the different intermediates which are actually being participating into the different types of other metabolic pathways. So what you see here is that the pyruvate, it is being converted into the acetyl CoA, and then the acetyl CoA, when it enters into the TCA cycle, it is actually producing the citrate. So citrate is actually being connected to the fatty acid and the sterol pathway. So that's how it can actually be able to communicate. So there will be a shuttling of the intermediates from the Krebs cycle to these pathways, and exactly the same way the intermediates could actually be shuttled back to the into the citrate cycle also. Then once the citrate is getting converted into the alpha ketoglutarate can directly be converted into the glutamate, and the glutamate, which is the amino acid actually, is can be shuttled back, and there are metabolic pathways so that the glutamate can be converted into the arginine, proline and glutamine, which means at this stage it actually can easily be able to communicate with the protein synthesis machinery. So you see the catabolic pathway is interacting with the anabolic pathways. And then we have the succinyl CoA. The succinyl CoA is a precursor of the porphyrin and the heme as well as the chlorophyll synthesis. So that's why it actually can shuttle the intermediates between the biosynthetic pathway of the heme biosynthesis. And once the succinyl CoA is getting converted into the malate, the malate is also getting shuttled between the with the pyruvate, and the enzyme is the pyruvate carboxylase and as well as the malic enzyme. Whereas the oxaloacetate what is being produced is actually can be converted into the phosphoenol pyruvate, or it is also can be converted into the another amino acid which is called as the aspartate or the other amino acids, and that's how, and once it can converted into the aspartate or the other amino acid, that also can enter into the pyridine or the pyrimidine pathway, which means it also can enter into the nucleic acid biosynthetic pathway. Uh, Nucleic acid biosynthetic pathways. So what you see here is that the Krebs cycle is a catabolic pathway, but it is actually participating with the different types of metabolic pathways, whether it is the fatty acid biosynthetic pathway, protein synthesis pathway, heme pathway, or nucleic acid biosynthetic pathway, or it is also can shuttle back to the phosphoenol pyruvate, and phosphoenol pyruvate is a precursor for the glycine, serine and cysteine pathway. So that's why and if there is a requirement, the Krebs cycle can also be able to produce the glucose, which can be stored in the form of the glycogen in the muscle cells. So what you see here is that the Krebs cycle has the very huge role in terms of the regulating the different types of metabolic pathways, and the intermediates actually can go into the different types of metabolic pathways. Like the lipid metabolism is connected to the Krebs cycle through the common intermediates such as the citrate and the acetyl CoA. Similarly, the protein metabolism shares the intermediate as the alpha ketoglutarate, oxaloacetate. As a result, the Krebs cycle can allosterically or through the product inhibition regulates other metabolic pathways. In addition, it can redistribute the intermediates between the metabolic pathway and helps in the conversion of sugar to the protein, lipid, or the vice versa. And because of the such a very high level of role in the metabolic pathways, the Krebs cycle is been taking place very high in terms of the evolutions. So what is the role of the Krebs cycle in the evolution is that the Krebs cycle is directly associated with the running of electron transport chain, and hence depending on the availability of the oxygen. Development of the Krebs cycle has evolved the organism to adopt the high oxygen environment, which means earlier the people, like for example the prokaryotic cell, they don't have the Krebs cycle. Why they don't have the Krebs cycle? Because they are living in an environment where the oxygen availability is very low. So that's why they are happy with the glycolysis pathway, and they are only producing the very low amount of energy by the glucose molecules. But with the development of the Krebs cycle and with the adaptation of the Krebs, the mitochondria into the eukaryotic system, the Krebs cycle has made made it possible for the organism to adapt to the high energy environment, because in the high energy environment the Krebs cycle can be operated and it can actually be able to help the organism to produce the high amount of energy. So with this, we would like to conclude our lecture here. In our subsequent lecture, we are going to discuss more about the biomolecules. What we have discussed so far, we have discussed about the carbohydrate structure and functions, and we have also discussed how the carbohydrates are being utilized into the different types of metabolic pathways to produce the energy. And we have discussed about the glycolysis, we have discussed about the regulation of the glycolysis, and subsequent to that we have also discussed about the Krebs cycle and its regulations. And lastly, we have also discussed about the significance of the Krebs cycle into the different types of metabolic pathways. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss about some more biomolecules. Thank you.